The overall goal of this procedure is to better visualize stained fixed zebrafish embryos at early developmental time points. This is accomplished by first staining the zebrafish embryo using whole mount in situ hybridization. Next, an embryo is selected to flat mount and a central incision is made into the yolk with a pair of fine forceps. Then, the lash tools are used to gently scrape and remove the remaining yolk granules. Finally, the embryo is mounted in glycerol on a slide to enable the appropriate imaging of the specimen. Ultimately, flat mounting allows better visualization of stained embryos from a dorsal view by removing the yolk and positioning the embryo in a two-dimensional preparation.
The main advantage of this technique over existing methods like imaging intact embryos at younger developmental stages is that flat mounting removes the yolk sac, allowing the entire embryo to be viewed.
This method can help answer key questions in the developmental field, such as characterizing the domain of the renal-free gender field in an embryo.
Generally, individuals new to this method struggle because this technique requires delicate handling of the embryo, and also because learning the appropriate amount of pressure needed to remove the yolk sac can take time.
Demonstrating this procedure will be myself, Zoe, and Amanda, who are graduate students in the Wingert laboratory.
After collecting, fixing, and staining embryos according to the text protocol, select an embryo for flat mounting. With 1x PBST, transfer the embryo into a plastic or glass petri dish. Then, with the dish under a stereo microscope, use fine forceps to roll the embryo.